This podcast will deal with smooth muscle. Smooth muscle lacks striations and it lacks T-tubes. Smooth muscle shows involuntary contractions. The contractions in smooth muscle are regulated by the autonomic nervous system and by hormones. Smooth muscle can be very active metabolically and synthetically. Smooth muscle can synthesize exogenous proteins like collagen, elastin, various glycosaminoglycans, proteoglycans, and various growth factors. Smooth muscle is located in the walls of hollow viscera, so you'll find it in the organs like the GI tract or in the urogenital tract. Smooth muscle makes up the walls of blood vessels. You'll find smooth muscle in larger gland ducts. You'll find smooth muscle in respiratory passages and in the dermis of the skin. Now, smooth muscle cells have biosynthetic properties that are similar to fibroblasts, and some authors believe that smooth muscle cells might be part of a connective tissue family of cells. In other words, they're almost like an undifferentiated fibroblast-like cell, and some people have suggested that fibroblasts might actually be stem cells for smooth muscle cells. So in this cartoon, it's suggested that fibroblasts can be stem cells for osteoblasts, for chondroblasts, for adipocytes, and for smooth muscle cells. The point is that all of these cells are metabolically very active and they have some of the same metabolic characteristics. This is a histological view of smooth muscle. Smooth muscle, the cells are fusiform. They're elongated cells, longer than they are wide. They have extensive numbers of gap junctions between the cells. There's a central oval nucleus with several pronounced nucleoli. The nucleus in smooth muscle cells assumes kind of a corkscrew appearance as cells contract, and that corkscrew type appearance can actually be a useful diagnostic criteria when you look at smooth muscle cells in the laboratory. Each smooth muscle cell is surrounded by an external lamina or endomysium. Dense bodies in smooth muscle play a role in the contraction of myofilaments. The myofilaments interweave with each other, but they don't interweave in a regular repeating pattern. That's why you don't see the regular striations. The thin filaments in smooth muscle consist of actin and tropomyosin, but there's no troponin in smooth muscle. The thick filaments consist of myosin. If you focus on the red text on this slide, that's the way to compare and contrast smooth muscle with striated muscle. Let us back up for a minute. This is the bipolar thick filament in striated muscle with the M line. Now, I didn't mention this before, but why do we call it bipolar? You may remember as we looked at striated muscle, particularly skeletal muscle, we looked at the myofilaments, we looked at the thick filaments, and we saw them arranged so that there were these myosin heads on opposite ends to each other, and you might recall that these myosin heads bind onto the thin filaments and they cause the Z lines to come closer together as the striated muscle contracts. So there's a bipolar arrangement of thick filaments in striated muscle. But on the other hand, the thick filament in smooth muscle has more or less this side polar arrangement where you see these heavy meromyosin heads spanning the entire length of the thick filament as opposed to being on the ends of the thick filament as they would be in striated muscle. So a side polar arrangement where the heavy meromyosin heads span the length of the thick filament in smooth muscle. Here is a model that we use to talk about smooth muscle contraction. What's shown are the contractile filaments that contain actin and myosin, and then supporting fibers shown in the various shades of blue that contain various intermediate filaments. But these contractile myofilaments are not arranged in a regular array like they would be in striated muscle. And then there are densely staining plasma membrane attachment sites shown here, and cytoplasmic attachment sites shown like this. Dense membrane-associated attachment sites and dense cytoplasmic sites where the myofilaments interact with each other. So you have cytoplasmic dense bodies and membrane-associated dense bodies. The thick and thin myofilaments are not regularly arranged. Rather, they form this crisscross pattern with various intermediate filaments like vimentin and desmin. The thick filaments, which are myosin, line up so heavy meromyosin heads project from the entire length of the filament. So there's a much larger surface area for interaction of actin and myosin, and that's what this inset is trying to depict. Contraction of smooth muscle cells are very slow, they're very forceful, and they can be of long duration. And lest you think that this is not significant, let me remind you of the force of contractions as a woman is giving birth. The uterine contractions, probably some of the strongest contractile forces generated by any muscles in the body. So smooth muscle can have very forceful contractions, and they can be of relatively long duration. The all or none law for striated muscle contraction does not apply. So in a smooth muscle cell, the entire cell may contract, or only a portion of the smooth muscle cell may contract. This just highlights these densely staining plasma membrane attachment sites and the densely staining cytoplasmic sites. In the micrograph, they're the cytoplasmic dense bodies. This is the example diagrammatically of the smooth muscle cell contracting, and as it contracts, because you may not be getting all of the cell contracting at the same time, you'll see this squiggly shape to the nucleus. These cytoplasmic densities contain alpha-actinin, and here the red lines would be the actin and myosin filaments. So the contractile forces are harnessed by interaction of myofilaments with intermediate filaments at the membrane-associated and cytoplasmic dense bodies. It turns out that these membrane-associated and cytoplasmic dense bodies are analogous to the Z discs of striated muscle, because the intermediate filaments and actin attach to the dense bodies by alpha-actinin. So very much like you would expect to find in the Z discs of striated muscles. Here is how we believe a contraction occurs. Note that in the top cartoon, the myofilaments are not in a regular array. Rather, they're arranged obliquely to the long axis, and they're anchored onto the cytoplasmic and membrane-associated densities, which consist of desmin and vimentin. So here is what we think occurs. Here's a dense body with alpha-actinin. Here are the intermediate filaments, desmin and vimentin. Here's a membrane-associated dense body. Here are the thin filaments, like so in green, and the very long side polar arrangement of the thick filaments. So as the muscle is contracting, you can imagine the thin and thick filaments sliding past each other, but because they're anchored at all various points in the muscle cell is scrunching up. So these densities are analogous to the Z line of striated muscle, and they contain the actin-binding protein alpha-actinin. And so the contraction in smooth muscle causes the smooth muscle cell to assume this corkscrew appearance. Here's the fine structure of smooth muscle cells. There's actually portions of 3 smooth muscle cells here. So there's one smooth muscle cell like this, another smooth muscle cell like this, and a third smooth muscle cell like this third one showing part of the nucleus in the plane of section. Here is the basal lamina between two cells and the basal lamina between these cells, and you can see the cytoplasmic densities highlighted by the single and double arrows. Alpha-actinin and cytoplasmic dense bodies are in contact with the sarcolemma. That's shown here very nicely, and they appear to be highly branched structures as shown here in the cytoplasm of this particular cell. As we said, these nodal bodies or dense bodies are equivalent to the Z discs of striated muscle. There's a basal lamina. Smooth muscle cells lack a T-tubule system. Calcium from the extracellular matrix enters the smooth muscle cell through caveoli. Here is a higher power view of smooth muscle cells. Here's a portion of one cell, a portion of another cell, basal lamina here, basal lamina here. Look at these invaginations of the plasma membrane. You might imagine as these are invaginating, they're bringing calcium from the extracellular space into the cytoplasm. Now it turns out there's a calcium-calmodulin complex and a myosin light chain kinase that activates myosin so that actin can bind to it and cause smooth muscle cells to contract. And this diagram goes over what we think occurs in smooth muscle cells as they contract. There are calcium release channels that are in the cytoplasmic vesicles or caveoli where calcium is stored and released for the smooth muscle cell to contract. So here are these calcium release channels, maybe some in the membrane itself, but some in these vesicles that are associated with the ER. As a hormone binds or as the cell receives a signal to contract, activates a second messenger which opens these calcium release channels. The calcium is released into the cytoplasm, and a calmodulin, which is a calcium binding protein found in smooth muscle, binds to the calcium, and you have this calcium calmodulin complex. There is this myosin light chain kinase that activates the actin binding sites on the heavy meromyosin chains, and this causes binding to the thin filaments, the actin filaments, and there is contraction. What happens is myosin light chain kinase phosphorylates these light meromyosin chains in the myosin, and that's what's going to bind onto the actin when they're phosphorylated. A phosphatase splits this phosphate off, that inactivates the myosin, and the smooth muscle cell will then relax. Innervation of smooth muscle. The neuromuscular junction in smooth muscle is not as well organized as in skeletal muscle. There's sympathetic innervation with the neurotransmitter being norepinephrine. Parasympathetic innervation, the neurotransmitter is acetylcholine, and you'll probably talk about this in physiology. There's multi-unit innervation, so that every cell receives individual innervation, as, for example, in the sphincter pupillae of the iris, or in portions of the male genital tract. So the smooth muscle in the vas deferens shows multi-unit innervation. On the other hand, there can also be single unit, or what's called visceral innervation, where only a few cells receive innervation, and then the impulse for transmission for contraction goes to adjacent cells through the extensive gap junctions, and this is very common in the smooth muscle in the GI tract and in the urogenital tract. We will see histologically in the laboratory the myenteric plexus in the muscularis externa of the GI tract, for example.